హాయ్, ఇప్పుడు మన నెక్స్ట్ టాపిక్ సెల్ ద యూనిట్ ఆఫ్ లైఫ్ గురించి. సో ఈ టాపిక్లో మనం డీటెయిల్డ్గా సెల్ ద యూనిట్ ఆఫ్ లైఫ్, సెల్ సైకిల్ అండ్ సెల్ డివిజన్ నుంచి క్వశ్చన్స్ చూడబోతున్నాం. వెరీ ఫస్ట్ క్వశ్చన్, ఇఫ్ యూ రిమూవ్ ద ఫింబ్రియే ఫ్రమ్ ద బ్యాక్టీరియల్ సెల్, విచ్ ఆఫ్ ద ఫాలోయింగ్ వుడ్ యూ ఎక్స్పెక్ట్ టు హ్యాపన్? ఇప్పుడు బ్యాక్టీరియల్ ఎక్స్టెన్షన్లో మనకి ఫ్లెజెల్లా, పిల్లై, ఫింబ్రియే. ఫ్లెజెల్లా ఏంటి? మూమెంట్ లేదా లోకోమోషన్కి. అదే మనకి పిల్ల అయితే సెక్షువల్ రీప్రొడక్షన్కి, పిల్ల ఏమో సెక్షువల్ రీప్రొడక్షన్ అంటే కాంజుగేషన్కి. ఫింబ్రియాకి వచ్చేసరికి అటాచ్మెంట్ టు ద సబ్స్ట్రాటమ్ అండ్ రఫ్ సర్ఫేసెస్ అండ్ రాక్ మెటీరియల్స్. ఈ మొత్తం ఆన్సర్స్లో బాగా సూట్ అయ్యేది ద బ్యాక్టీరియా వుడ్ నాట్ అడ్హెర్ టు ద హోస్ట్ టిష్యూ ఆర్ సబ్స్ట్రాటమ్. బ్యాక్టీరియా సబ్స్ట్రాటమ్కి కానీ హోస్ట్ టిష్యూస్ కానీ అతుక్కోదు ఫింబ్రియా లేకపోతే. విచ్ వన్ ఆఫ్ ద ఫాలోయింగ్ ఈజ్ నాట్ ఎ గుడ్ కన్స్టిట్యూయెంట్ ఆఫ్ సెల్ మెంబ్రెయిన్? ఈజ్ నాట్ ఎట్ ఆల్ ఏ కన్స్టిట్యూయెంట్ ఆఫ్ సెల్ మెంబ్రెయిన్. సెల్ మెంబ్రెయిన్లో అసలు లేనిదేది? సెల్ మెంబ్రెయిన్లో లేనిది స్టార్చ్. కొలెస్ట్రాల్, ప్రోటీన్స్, లిపిడ్స్, ఫాస్ఫోలిపిడ్స్ ఉంటుంది, స్టార్చ్ ఉండదు. సో ఆప్షన్ 3 రైట్. సో వేర్ ఆర్ ఎక్స్క్రీటరీ ప్రోడక్ట్స్ ఆర్ అదర్ అన్వాంటెడ్ మెటీరియల్ స్టోర్డ్ ఇన్ ద ప్లాంట్ సెల్? సో ఎక్కడ స్టోర్ అవుతుంది? వాక్యూల్స్లో స్టోర్ అవుతుంది. వాక్యూల్స్లో మనకి టోనోప్లాస్ట్ ఉంటుంది, ప్లస్ సెల్ సాప్ ఉంటుంది. సో అగెయిన్స్ట్ కాన్సన్ట్రేషన్ గ్రేడియంట్ అంటే సైటోప్లాజం కంటే కూడా లోపల హైపర్ టోనిసిటీ మెయింటైన్ అవుతుంది కాబట్టి లోపల మనం ఎక్స్ట్రా ఎఫర్ట్తో పుష్ చేయాలి, అంటే యాక్టివ్ ట్రాన్స్పోర్ట్ జరుగుతుంది. సో ఆప్షన్ 3 ఈజ్ రైట్. నెక్స్ట్, విచ్ ఆఫ్ దీస్ ఈజ్ నాట్ ఎ ఫంక్షన్ ఆఫ్ గోల్గి ఆపరేటస్? గోల్గి ఆపరేటస్ ఫంక్షన్ కానే కాదు ఇది. సైట్ ఆఫ్ ఫార్మేషన్ ఆఫ్ గ్లైకోప్రోటీన్స్ ఇంకా గ్లైకోలిపిడ్స్ కరెక్టే. క్రియేషన్ ఆఫ్ సెక్రీటరీ వెసికల్స్ కరెక్టే. Packing of materials meant for intracellular and extracellular delivery. So correct. So and next, protein synthesis beginning from amino acids. No, adi chesedi RER. Non-membrane bounded cell organelle in Mangifera indica. అంటే ప్లాంట్ సెల్లో నాన్ మెంబ్రెయిన్ బౌండెడ్ సెల్ ఆర్గనల్ ఏది? రైబోజోమ్స్. రైబోజోమ్స్ చుట్టూ ఎటువంటి మెంబ్రెయిన్ ఉండదు. కెరోటినాయిడ్ పిగ్మెంట్స్ ఆర్ ఫ్యాట్ సాల్యుబుల్. ఎందుకంటే కెరోటిన్ ఈజ్ ఫ్యాట్ సాల్యుబుల్ వైటమిన్ కదా. సో రెటినాల్ ఇస్ అ ఫ్యాట్ సాల్యుబుల్ వైటమిన్. అది దేంట్లో నుంచి వస్తుంది? కెరోటిన్ నుంచి వస్తుంది. సో అలా గుర్తుపెట్టుకోండి. కెరోటినాయిడ్ పిగ్మెంట్స్ ఆర్ ఫ్యాట్ సాల్యుబుల్. ద చీఫ్ రోల్ ఆఫ్ న్యూక్లియోలస్ ఇన్ ద న్యూక్లియస్ ఈజ్, న్యూక్లియోలస్ ఏం చేస్తుందంటే రైబోసోమ్స్కి కావాల్సిన ఆర్ఎన్ఏని ప్రొడ్యూస్ చేస్తుంది. సో ద కరెక్ట్ ఆన్సర్ ఈజ్ రైబోజోమల్ ఆర్ఎన్ఏ సింథసిస్, ఆప్షన్ 2. 108. సిస్టర్నే అంటే గోల్జీ బాడీస్లోని వెసికల్స్, ట్యూబ్యూల్స్ సిస్టర్నే అంటాం. వాటి డయామీటర్ ఎంత? 0.5 to 1 micrometer. So 1 is the right answer. You would expect a cell with an extensive Golgi apparatus too. దేనికోసం? ప్యాక్ మెటీరియల్స్, ఆప్షన్ 3 ఈజ్ రైట్. మెటీరియల్స్ని ప్యాక్ చేయడానికి. సో మెటీరియల్స్ని ప్యాక్ చేయడం అంటే లిపిడ్స్ని, ప్రోటీన్స్ని ప్రొడ్యూస్ చేస్తే దాన్ని గ్లైకోలిపిడ్స్, గ్లైకోప్రోటీన్స్గా గోల్జీ బాడీస్ ప్యాక్ చేస్తాయి. Neutral solutes may move across the membrane by the process of. న్యూట్రల్ సొల్యూట్స్ డిఫ్యూజన్ ద్వారా వెళ్తాయి. ఓకే, సో రిమెంబర్ న్యూట్రల్ సొల్యూట్స్ డిఫ్యూజన్. పొలారిటీ ఉంటేనే మనకి ఇంకేదన్నా ఆల్టర్నేటివ్ ఉంటుంది. సో న్యూట్రల్ సొల్యూషన్ కాబట్టి డిఫ్యూజన్. సో 111. చూస్ ద ఇన్కరెక్ట్ స్టేట్మెంట్ రిగార్డింగ్ సెల్ మెంబ్రెయిన్. సెల్ మెంబ్రెయిన్ గురించి ఒక ఇన్కరెక్ట్ స్టేట్మెంట్ చూస్ చేసుకోమన్నారు. Lipids of the membrane have a quasi-fluid nature, allows the lateral movements of protein. Correct. Fluidity of the membrane allows for important process like cell division, endocytosis, etc. to take place easily. Correct. Carrier proteins helps in transport of polar molecules across the membrane. Idi kuda correct. Neutral solutes cannot undergo simple diffusion across the membrane. Can undergo simple diffusion. Wrong statement ఇది, ఆప్షన్ 4. న్యూట్రల్ సొల్యూట్స్ కెన్ అండర్గో సింపుల్ డిఫ్యూషన్. యూనిసెల్యులర్ ఆర్గానిజమ్స్ దేనికి? క్యాపబుల్ ఆఫ్ ఇండిపెండెంట్ ఎగ్జిస్టెన్స్ అండ్ కెన్ పర్ఫార్మ్ ఎసెన్షియల్ ఫంక్షన్స్ ఆఫ్ లైఫ్. ఆప్షన్ 3 ఈజ్ రైట్. ఓకే, క్యాపబుల్ ఆఫ్ ఇండిపెండెంట్ ఎగ్జిస్టెన్స్ అండ్ కెన్ పర్ఫార్మ్ ఎసెన్షియల్ ఫంక్షన్స్ ఆఫ్ లైఫ్. విచ్ ఆఫ్ ద ఫాలోయింగ్ స్టేట్మెంట్స్ ఆర్ కరెక్ట్ ఫర్ ఎ గివెన్ స్ట్రక్చర్? సో ఇది స్మాల్ స్ట్రక్చర్, ఇట్ హ్యాస్ 9+2 అరేంజ్మెంట్. ఇది సీలియా లేదా ఫ్లెజెల్లా. దీనికి 9+2 అరేంజ్మెంట్ ఉంటుంది, రైట్. ఇట్ ఈస్ కవర్డ్ విత్ ప్లాస్మా మెంబ్రెయిన్, రైట్. ఇట్స్ కోర్ ఈస్ కాల్డ్ axoneme, రైట్. ఆల్ ఆఫ్ దిస్. So all of this is the right answer. Cis ఇంకా trans phases of Golgi apparatus are entirely different but interconnected. This is an NCERT phrase. Remember, the entirely different but interconnected cis and trans phases of Golgi apparatus. Size of typical eukaryotic cell. This is also from NCERT line. 10 to 20 micrometers is the size of typical eukaryotic cell. Helicobacter pylori are bacteria that are chronically infect stomach. Human stomach ni infect chestadi. Severe gastric ulcers ni తీసుకొస్తుంది. Which bacterial cell part helps these bacteria to remain attached to the host cell against churning action of the stomach wall, causing contents to mix vigorously. ఏ స్ట్రక్చర్ అటాచ్ అయ్యేలా చేస్తుంది అంటే ఫింబ్రియే. సో ఆప్షన్ 3. ప్రోకారియోటిక్ సెల్స్ ఆర్ జనరల్లీ స్మాల్ అండ్ మల్టిప్లై ఫాస్టర్. ఆప్షన్ 3 ఈస్ కరెక్ట్ దాన్ యూకారియోటిక్ సెల్స్. విచ్ ఆఫ్ దీస్ ఈజ్ నాట్ కరెక్ట్ రిగార్డింగ్ ATS రైబోసోమ్స్? ATS రైబోసోమ్స్ గురించి కరెక్ట్ కాని పాయింట్స్ ఏవి? డు నాట్ హ్యావ్ membranes of their own. వాటికి మెంబ్రెయిన్స్ లేవు. present in the cytoplasm on RER of prokaryotes. అసలు రఫ్ ఎండోప్లాస్మిక్ రెటిక్యులం ప్రోకారియోటిక్స్లోనే ఉండదు. సో దిస్ ఈస్ ద రైట్ ఆన్సర్. సో దిస్ ఈస్ నాట్ కరెక్ట్ రిగార్డింగ్ ATS. రిమైనింగ్ ఆల్ ఆర్ రైట్. దే ఆర్ యాబ్సెంట్ ఇన్ క్లోరోప్లాస్ట్ అండ్ మైటోకాండ్రియా, ఎందుకంటే 70S రైబోసోమ్స్ ఉంటాయి కాబట్టి. దే టేక్ పార్ట్ ఇన్ ప్రోటీన్ సింథసిస్, రైట్. ద టైప్ ఆఫ్ రైబోసోమ్స్ ఫౌండ్ ఇన్ కోకోనట్ లీఫ్ క్లోరోప్లాస్ట్. క్లోరోప్లాస్ట్ లోపల ఏముంటుంది? క్లోరోప్లాస్ట్ లోపల 70S రైబోసోమ్స్ ఉంటాయి. సో S stands for Swedberg. So S stands for sedimentation coefficient. S refers to Swedberg. Okay, S refers to Swedberg and S stands for sedimentation coefficient. So 70S, 50S plus 30S. Option 2 is right. Fluid mosaic model was proposed in the year 1972. సో ఆప్షన్ 3 1972లో ప్రపోజ్ చేశారు. విచ్ ఆఫ్ ద ఫాలోయింగ్ ఈజ్ ద లార్జెస్ట్ కన్స్టిట్యూయెంట్ ఆఫ్ మెంబ్రెయిన్ ఆఫ్ ద ఎరిథ్రోసైట్స్ ఇన్ హ్యూమన్ బీయింగ్స్ అండ్ ఈజ్ ఆల్సో రెస్పాన్సిబుల్ ఫర్ ఫార్మింగ్ మోస్ట్ ఆఫ్ ద ఫంక్షన్స్ ఆఫ్ ద మెంబ్రెయిన్? ప్రోటీన్స్ 52% ప్రోటీన్స్ ఉంటాయి, 40% లిపిడ్స్ ఉంటుంది. ఏ పాలీపెప్టైడ్ హ్యాస్ బీన్ మేడ్ ఇన్ RER. It is to be transported to the Golgi body to be converted into glycoprotein. Yes, RER ప్రొడ్యూస్ చేసిన ప్రోటీన్ని Golgi బాడీకి పంపిస్తే అక్కడ గ్లైకోప్రోటీన్ అవుతుంది. విచ్ పార్ట్ ఆఫ్ Golgi బాడీ ఫస్ట్ రిసీవ్ ద పాలీపెప్టైడ్ ఫ్రమ్ RER? సిస్ అంటే కాన్వెక్స్. సిస్ ఫేజ్ అంటే కాన్వెక్స్. ఇది కొంచెం కన్ఫ్యూజింగ్ ఉంటుంది అందరికీ, కానీ సిస్ అంటే కాన్వెక్స్, ట్రాన్స్ అంటే కాన్కేవ్. సో సిస్ కాన్వెక్స్ రిసీవ్ చేసుకుంటుంది. ఆప్షన్ 1 ఈజ్ రైట్. సో రీడ్ ద గివెన్ స్టేట్మెంట్స్. సెంట్రోమియర్ ఈజ్ ప్రెజెంట్ ఇన్ ద మిడిల్ ఆఫ్ క్రోమోసోమ్ అండ్ ఫార్మ్స్ 2 ఈక్వల్ ఆర్మ్స్. దీనికి వచ్చి మెటాసెంట్రిక్ 1. సెంట్రోజోమ్ హ్యాజ్ ఎ టర్మినల్ క్రోమోజోమ్ హ్యాజ్ ఎ టర్మినల్ సెంట్రోమియర్. దీనికి ఆప్షన్ టీలోసెంట్రిక్కి 2 ఉండాలి. Centromere lies close to the end of the chromosome, forming one extremely short and one very long. దీనికి acrocentric. And next ఫైనల్గా వచ్చి centromere is slightly away from middle, resulting in one shorter and one longer. So option 1 is matching. Metacentric, submetacentric, acrocentric, telocentric. So 124 match చేయాలి. A is centromere, right? C is secondary constriction, B satellite. So option 3, centromere, satellite, secondary constriction. The cementing layer of calcium pectate between adjacent plant cells ni ఏమని పిలుస్తాం? middle lamella, which is made up of calcium pectate. So cell plate ఫ్యూచర్లో middle lamella అవుతుంది. So a single human cell has approximately 2 meters of long thread of DNA distributed among its 46 chromosomes. So option 1 is right. Categorize the given statements as true or false. Mitochondrial membranes have their own specific enzymes for the functions of its mitochondria. True. Euglenoid shows the presence of plastids. True. Without staining, mitochondria is not easily visible under the microscope. True. Chloroplast stroma doesn't have enzymes for the manufacture of proteins. False. They have enzymes Yes, that is right. So for the manufacture of proteins, they don't have enzymes. Yeah, so that is the false statement. They have the enzymes. Where is the luminal part of cell is seen? So luminal, extraluminal means inside ER and outside ER. So luminal means inside ER. So that means inside ER. Option 4 is right. Mesosomes are infoldings of plasma membrane, which is found in prokaryotes. Analogous structure for mitochondria. Origin is different, but they both work for respiration. Sodium and potassium pump is an example of active transport. Option 1 is right. Which of the following is an incorrect match? So starch storage of potato tuber is amyloplast, right? Oil storage within the cells of groundnut, eleoplast, right? Fat storage in the cells of adipose tissue, eleoplast. Adipose tissue is not seen in plants, it is seen in animals, so it's wrong. So this is the incorrect one. And starch storage in sweet potato, amyloplast, this is right. The common characteristic feature of a plant sieve tube cells and most of the mammalian erythrocytes is. So that is absence of nucleus. Option 4. They both doesn't have the nucleus. Which one of the following statements is wrong for nucleolus? It is not a membrane bounded structure, right? It is a spherical structure found in nucleoplasm, right? It is the site for the synthesis of active ribosomal protein. This is wrong. Active ribosomal RNA, not the protein, right? So option 3 is appropriate. This is the wrong statement for nucleolus. The outer membrane of nucleus is continuous with endoplasmic reticulum, right? Do not bear ribosomes. It bear ribosomes. So statement 1 is true and statement 2 is incorrect. So only statement 1 is correct. Option 1. Bacteria can be classified into 2 groups based on the differences in cell envelope, gram positive and gram negative. Which of these cellular parts are not seen in any of the animal kingdom? Plastids. Plastids are seen only in plant kingdom. Number of chloroplasts in the mesophyll cells is 20 to 40 per cell. How wide is the perinuclear space? 10 to 50 nanometers. So option 2, 10 to 50 nanometers, it is given in the NCERT also. Statement 1, 9 plus 2 array is observed in cilia, right? 9 plus 0 array is observed in flagella, wrong. 9 plus 2 only it's also. 9 plus 2 array is observable in centriole, this is also wrong. 9 plus 0 is observed in centriole. So only statement 1 is correct. Remaining 2 are wrong. So option 3 is more appropriate. Cell membranes can show which of these biomolecules? Phospholipids, cholesterol, carbohydrates and proteins. All the 4 can be seen. And next, 141. Study the following statements regarding mitochondria and select the incorrect one. These are the sites of aerobic respiration, right? Matrix contain 10 to 20 circular DNA, double-stranded DNA molecules, few RNA molecules and 70s ribosomes. Mitochondria will be dividing by fission. This is right. The number of mitochondria is a constant for every cell depending on the species. No. So 2 and 4 are wrong. So they don't have 10 to 20 double stranded. And next, select the incorrect statement. Robert Brown discovered the nucleus, right? Robert Hooke first saw and described living cell. This is wrong. Dead cell. So option 2 is appropriate. Plastids are present in all plant cell and euglenoids. So both 1 and 2. In a eukaryotic cell, in which of the following functions is the cytoskeleton is not involved? For cell wall synthesis. For cell wall synthesis, cytoskeleton is not involved. In animal cell, lipid like steroid hormones are synthesized in SER, smooth endoplasmic reticulum. Chromatin is made up of DNA plus basic proteins called histones. Option 1 is right. The tail of phospholipid molecule is hydrophobic and it is composed of saturated hydrocarbons. This is the NCERT phrase. Option 3 is more appropriate. Every chromosome has a primary constriction called centromere. Option 2 is correct. Considering that the primary wall is yet to disappear in the plant cell, while the secondary wall is already created, the following layers are present near the plasma membrane in the plant cell. Near the plasma membrane will be secondary wall. Primary wall, secondary wall, tertiary wall, plasma membrane. Cell organelles that can be seen in cells belonging to both Plantae and Animalia: microbodies, Golgi bodies, ER, mitochondria. Plastids are seen only in kingdom Plantae. So option 4. Pick out the correct statements. So all the questions are regarding cell cycle and cell division. Let's see. A mother cell with 40 chromosomes can undergo mitosis, right? A mother cell with 20 chromosomes can undergo mitosis, right? A mother cell with 39 chromosomes can undergo mitosis, which is 2n-1 aneuploidy, right? The person born with Down syndrome will be forming the whole body with mitosis, right? So 1, 2, 3, all the 3 are right. What are asters with respect to cell division? Asters are nothing but a pair of centriole radiating microtubules at the poles of mitotically dividing cells. Option 1 is correct. A picture of dividing potato cell taken through the microscope shows that the cell contains 48 chromosomes. Each consists of 2 chromatids. The picture must have been taken as metaphase of mitosis. So 48 chromosomes consisting of 2 chromatids. Anaphase will be splitting. And next, in meiosis during prophase 1, recombination nodule is observed during Pachytene, because the crossing over will take place during Pachytene. So option 4. And recombinase enzyme also will be acting for exchange of the non-sister chromatids. Chromosomal number is halved in meiosis during anaphase 1. Option 2 is appropriate. Read the following statements about the cell division and select the correct ones. M phase represents the phase where actually cell division occurs, and one phase represents the phase between, and the interface represents the phase between 2 successive M phases, right? In the 24 hours average duration of cell cycle of human cell division lasts for only about an hour, right? M phase constitutes 95% of the duration of cell cycle. Wrong. Interphase constitutes 95% of the cell cycle. That is right. So 1 and 2 are correct. So 1 and 2. Option 1. A zygote is formed with odd number of chromosomes in humans from an abnormal ovum of 24 chromosomes and a normal sperm of 23 chromosomes. Which of the following is possible for this cell and an individual produced from this zygote? The cell can undergo normal mitosis. An individual can undergo both mitosis and meiosis. No. So the cell may undergo mitosis, but the individual cell can undergo only mitosis, not the meiosis. Option 2 is right. Aneuploidy people will not produce gametes because they can't make the meiosis. So option 2 is right. During cell division, spindle fibers get attached to the proteinaceous disc on the condensing chromosome, which we call it as kinetochore. Option 4 is correct. The pair of synapsed homologous chromosomes are called bivalent. Option 4 is appropriate. Crossing over is the exchange of genetic material between the 2 sister chromatids of the chromosome. No, between 2 non-sister chromatids. Statement 1 is false. Crossing over is done with the help of replication nodules that comprise the enzyme DNA polymerase. This is also wrong. Recombinase. So statement 1 and statement 2 are wrong. Both the statements are false. So option 2. A cell that is entering the M phase of mitotic cell cycle is either haploid or diploid, always with duplicated chromosomes. Option 2 is right. So there is a figure shown which is prophase 1. You have to find out at the end of meiosis 1 and meiosis 2 which picture is more appropriate. At the end of meiosis 1, second one is more appropriate. At the end of meiosis 2, third one is more appropriate. So 2 and 3. Option 2 is right. 163. Which of these is not correct about cytokinesis in plant cell? Occurs in the formation of cell plate. Yes, right? Cell plate represents the cell wall. No, cell plate represents the middle lamella, not the cell wall. So this is the right answer. Option 2 is not correct. Synaptonemal complex helps in synapsis of homologous chromosomes. Option 2 is correct. The cell with chromosomal number 2n is equal to 6 is in metaphase with all metacentric chromosomes with respect to the number of each of these in the cells arrange the descending order. So centrioles is 4. So centrioles is 4. Kinetochores is 12. Metaphasic plate is 1. Centromeres is 6. Arms of chromosomes is 24. Pole is 2. So we have to go with the descending order. So last we will be getting metaphasic plate because this is 1. So 3 has to be the last. So this is the more appropriate one. We have the very first one is arms of chromosomes 24 and kinetochore 12, right? And centromeres is 6. Centrioles is 4. And metaphasic plate is 1, right? Sorry, poles is 2. Later on 1. Tetrad consists of 2 homologous chromosomes with each with 2 chromatids. Option 4 is appropriate. Which of the following is the correct sequence of cell cycle of an epithelial cells that is just formed from the mother cell and will further divide to form a daughter cell? G1 S, G2 M. Option 3 is appropriate. The material commonly used in the laboratory for studying the mitosis process is onion root tip. Option 3 is correct. So the given diagram shows a cell. Which of the following statements related to the image is not correct? Nuclear envelope is disappearing. No, nuclear envelope is appearing. This is actually telophase. Cell is furrowing. Means after telophase it is going for cytokinesis of an animal cell. So option 1 is correct. This is not the correct regarding the diagram. It is an animal cell. It is in telophase. All are right. The significance of meiosis lies in reduction of diploid number of chromosomes to haploid, right? Conserving the constancy in chromosomal number in sexually reproducing organisms, right? Production of genetic variability in the population of the species, right? All of this, option 4 is right. A cell in G1 has 28 chromosomes and with total of 2 picograms of DNA. At the end of G2, what will be the chromosomal number and the DNA content of the same cell? Same 28 chromosomes, but DNA content will be doubled in the S phase. That will become 4 picograms of DNA. Mitotic anaphase differs from metaphase in possessing each pole. Differs from metaphase. Anaphase differs from metaphase in possessing each pole. Same the number of chromosomes with half the number of chromatids. Option 1. So cells of root tip of an onion has 16 chromosomes. How many chromosomes will be the cell is having at G1, S and in metaphase? 16, 16, 16. All are 16. During synapsis, the number of DNA molecules in the bivalent is 4. So bivalent means tetrad. Means each chromatid have one DNA molecule. So we have tetrad means 4 chromatids. So 4 DNAs. 175. B is anaphase, not metaphase. C is cytokinesis, not karyokinesis. A is M phase and D is synthetic phase. This is right. Option 3 is right. The duplication of centrioles occurs during interphase. Option 4 is right. 177. Disintegration of nuclear membrane. So disintegration of nuclear membrane is prophase. 1 is Q. Appearance of nucleolus. Telophase. R. Division of centromere. Anaphase. P. So option 1 is right. Replication of DNA S phase. Option 1 is right. At the end of meiosis in humans, the daughter cell receives a mixture of maternal and paternal chromosomes. Option 2. When a number of chromosomes are ready is already reduced to half in meiosis 1, what is the necessity of meiosis 2? So it requires the separation of chromatids of each chromosome and ultimately produce 4 daughter cells. Which stage is the connecting link between meiosis 1 and meiosis 2? Interkinesis without DNA replication and with interkinesis without DNA replication but with duplication of centrioles. Option 3 is right. Meiosis has an evolutionary significance because it results in recombination. Option 4 is appropriate. Which of the following statements is correct regarding G0? Cells at this stage are metabolically active, but their cell division is suspended temporarily or permanently. Option 4 is correct. Which one of the following occurs in the phase just before the occurrence of the phase, involving the reformation of nuclear envelope during M phase of cell cycle at apical meristem of bean root? So that is movement of chromatids of the opposite pole. In the question they are talking about telophase. So before telophase we have anaphase, movement of chromatids to the opposite poles. This is the right answer. In S phase, duplication takes place and it has started moving towards poles in prophase. We are talking about which component of which cell component? Centriole. Option 3 is right. Assembly of mitotic spindle. So we have to match it. Sorry, assembly of meiotic spindle. Duplication of centriole, S phase. So 2 is 5. 2 is V. Assemblage of meiotic spindle is diakinesis. 1 is U, 1 is U, 2 is V. Action of recombinase is packitine. 3 is S. And next, movement of chromatids towards opposite poles. Anaphase 2, T. So option 2 is appropriate for this. Next, section B. Which of the following statements are correct for cell cycle? So cell cycle is the sequence of events involving growth and division of the cells from the time of its formation to its own division into the daughter cells. Right? Cell growth in terms of cytoplasmic increase is a continuous process, right? DNA synthesis occurs only during specific stage of cell cycle, right? Replicated chromosomes are distributed to the daughter nuclei during cell division. So 1, 2, 3, 4 all are right. Mitotic spindle is mainly composed of tubulin fibers, tubulin protein, right? Read the given events occurring in mitosis. Select the correct sequence of the occurrence. Movement of centrioles towards opposite poles, right? Disappearance of nucleolus and nuclear membrane. So we have to align this. First one is 2. So 1, movement of centrioles towards opposite poles. Disappearance of nucleolus. 2, disappearance of nucleolus and nuclear membrane. Movement of centrioles towards opposite poles. Alignment of chromosomes at the equator. Separation of 2 chromatids of the centromere. Condensation of chromosomes to chromatin material. So exact sequence is option 1. If you provide with the root tip of onion in your class and ask the students to count the chromosomes, which is the following stage would you most convenient look into, and what will be the number of chromatids in this stage? It is metaphase. Metaphase, and it is 16. So option 1. Number of chromatids will be 32 in this stage. So in the 16 chromosomes we have 32 chromatids. In some, human cheek cells are undergoing prophase. The number of chromatids, chromosomes and kinetochores are respectively. The number of chromatids are 92. Chromosomes are 46. Kinetochores are 92. So option 2 is correct. PQ are the two stages of mitosis identified. P is prophase, Q is anaphase, option 4. Prophase and anaphase, right? 192. Which of the following identify the meiotic stage which simultaneously splitting of centromere and each of the chromosome takes place? Anaphase 2, splitting of centromere. Gametes have a genetic variation due to meiosis. Which one option is given below? Meiosis provide an opportunity for variation. The beginning of diakinesis is marked by terminalization. No. The two members of each bivalent seem to repel each other and move towards the opposite poles. Large recombination nodules appear at the chromosome of bivalent. So option 4 is right. The cell which enter dash phase differing into specific type of cells, Z0. Disjunction of members of homologous chromosomes in meiosis occurs during anaphase 1, option 2. Sherry prepared a slide of onion root tip. She created an image of the view under the microscope in her book. Which of the cells shows the correct identification of stages? So prophase is C. No. This is anaphase. Metaphase is E. Right. Metaphase is E. Right. C is telophase. No. This is anaphase. Anaphase is A. This is interphase. So option 2, metaphase is E, right. What would be the change in the chromosomal number during S phase? No change in the chromosomal number. Option 1 is right. 198. So you have to match the column. Correct answer. Prophase 1, crossing over occur, right. Metaphase 1, sister chromatids migrate to the opposite poles. No. Anaphase 1, homologous chromosomes line up at the equator, wrong. So A is correct, B and C are false. Option 3 is correct. So stages of cell division was incorrectly listed by a student in the order given below. Reorder the stages of mitosis, correct sequence of occurring and choose the correct option. So first one will be 2, 4, 3, 1, 5. Option 2. 2, 4, 3, 1, 5. Interphase. 2 is interphase, 4 is prophase, 3 is metaphase, 1 is anaphase and 5 is telophase. Select the correct statement. Chromosome decondense and lost their individuality during anaphase. Right? That is right. Spindle fiber attaches to kinetochore of chromosomes during early prophase. No, during late prophase. So centriole begin to move to the opposite pole during metaphase. No, during prophase also itself. DNA is replicated during S phase of the cell cycle. Sorry, first statement is wrong, second statement is wrong, third statement is wrong, only fourth statement is correct statement. Sorry, chromosome decondense and lost their individuality during telophase. So not anaphase. So option 4 is appropriate. DNA is replicated during S phase of cell cycle. So these are the questions from this chapter of cell, the unit of life, and cell cycle and cell division. I hope you got some of the important points from this chapter. Thank you very much. Bye bye.